This procedure uses the Virochip, a Pan-Viral Microarray Assay, to analyze clinical samples for both known and unknown viruses. First, nucleic acids are extracted from clinical samples. Next, the extracted RNA is reverse transcribed using random primers, second-strand cDNA synthesis is performed, and the cDNA library is amplified with random PCR. Then, the amplified cDNA is labeled with fluorescent dyes and hybridized to a Virochip microarray. The final step is to scan and analyze the microarray. Ultimately, results can be obtained that show the presence or absence of a virus or several viruses through analysis of the Virochip microarray.
Broad-spectrum detection methods such as a Virochip microarray are needed in diagnostic microbiology. Because common clinical syndromes such as pneumonia are not specific to any individual pathogen, and because of the continual emerging threat of novel viral pathogens such as the SARS coronavirus and 2009 novel H1N1 influenza virus.
This method can help answer key questions in the field of diagnostic microbiology, such as what percentage of undiagnosed infections in the clinical setting may be caused by as yet unidentified novel viruses. The implications of this technique extend toward better diagnosis of acute clinical illness in the hospital, because we are testing for hundreds to thousands of potential pathogens simultaneously.
We first had the idea for this method in 2002, when we decided to apply the newly developed technology of microarrays, pioneered in the laboratory of Dr. Joseph DeRisi at UCSF, to the detection of viruses. Our first real-world test case was using the Virochip to identify a novel coronavirus responsible for SARS in 2003. Demonstrating the procedure from start to finish will be a technician from my laboratory who will demonstrate the use of the Virochip assay on a nasal swab sample from a child to diagnose the cause of her acute respiratory illness.
Nasopharyngeal swab samples are collected in sterile containers containing viral transport liquid media and stored frozen in a -80°C freezer. In a biosafety hood, allow the tube containing the sample to thaw and briefly stir the swab in the media before disposing of the swab in a biohazardous waste bin. Then, aliquot 200 microliters of the sample into a 1.5 milliliter tube. Pass the sample through a 0.22 micron spin filter to purify for viral particles and into a fresh 1.5 milliliter tube. Next, use the Turbo DNase kit from Ambien, per manufacturer's instructions, to degrade host genomic DNA and purify for protected, encapsulated viral nucleic acid. Follow this with RNA extraction, using the Zymo-ZR Viral RNA Extraction Kit or QIAGEN Ultrasense Virus Kit per the manufacturer's instructions. Measure the concentration of the extracted RNA using a nanodrop nucleic acid spectrophotometer. RNA concentration is preferably at least 10 nanograms per microliter, but will vary from sample to sample. Using a thin-walled 200 microliter tube, add 1 microliter of primer A at a concentration of 40 picomoles per microliter to 4 microliters of extracted RNA. Heat the sample to 65°C for 5 minutes in a thermocycler, and then let the tube cool to room temperature for 5 minutes. Next, add 5 microliters of a master mix containing 2 microliters 5 times RT buffer, 1 microliter 12.5 millimolar DNTP, 1 microliter water, 0.5 microliters 0.1 molar DTT, and 0.5 microliters superscript 3 reverse transcriptase. In a thermocycler, incubate the tube at 42°C for 60 minutes. Then, heat the sample to 94°C for 2 minutes to abort the reverse transcriptase reaction. Cool to 10°C for 2 minutes and pulse centrifuge for 5 seconds. Next, add 5 microliters of sequenase mix, consisting of 1 microliter 5 times sequenase buffer, 3.8 microliters water, and 0.15 microliters sequenase. For second strand synthesis, ramp the thermocycler up from 10 degrees Celsius to 37 degrees Celsius over 8 minutes. Hold at 37 degrees Celsius for 8 minutes, then heat to 94 degrees Celsius for 2 minutes to inactivate the sequenase and cool to 10 degrees Celsius. At this point, if desired, store the sample containing 15 microliters cDNA at minus 20 degrees Celsius. First, add 5 microliters of cDNA sample to 45 microliters of a master mix consisting of 5 microliters 10 times PCR buffer, 1 microliter 12.5 millimolar DNTP, 1 microliter 100 picomole per microliter primer B, 1 microliter Clentac LA, and 37 microliters of water. Then, run the PCR protocol as follows: 94 degrees Celsius for 2 minutes, followed by 25 cycles of 94 degrees Celsius for 30 seconds, 50 degrees Celsius for 45 seconds, and 72 degrees Celsius for 1 minute. Then, an annealing step of 72 degrees Celsius for 5 minutes, followed by a hold at 10 degrees Celsius. After PCR, check the PCR product on a 1.5% agarose electrophoresis gel. A smear from approximately 200 to 1000 base pairs should be visualized. To incorporate AADNTP, add 5 microliters of viral PCR product to 45 microliters of a master mix consisting of 5 microliters 10 times PCR buffer, 1 microliter 12.5 millimolar AADNTP, 1 microliter 100 picomole per microliter primer B, 1 microliter Clentac LA, and 37 microliters water. Run the PCR protocol as follows: 94 degrees Celsius for 2 minutes, 15 cycles of 94 degrees Celsius for 30 seconds, 50 degrees Celsius for 45 seconds, 72 degrees Celsius for 1 minute, an annealing step of 72 degrees Celsius for 5 minutes, and hold at 10 degrees Celsius. Then, clean the new PCR product with the DNA Clean and Concentrator 3 kit per the manufacturer's instructions and dilute in 10 microliters of elution buffer. Add 1 microliter of 1 molar bicarbonate and 1 microliter Cy3 to the 10 microliters of PCR product and incubate for 1 hour in the dark. Next, again clean the Cy-labeled sample with DNA Clean and Concentrator 5 kit and dilute in 12 microliters of elution buffer. Use 1.5 microliters of sample to check cDNA concentration and the amount of dye incorporated on a nanodrop spectrophotometer. In a PCR strip tube, add 1 microliter 25 times fragmentation buffer, 5 microliters 5 times blocking buffer, 9.5 microliters, or amount necessary for a normalized final concentration of 10 picomoles per microliter of Cy3 labeled sample, and water to a total volume of 25 microliters. Then, add 25 microliters of 2 times GX hybridization buffer and spin down briefly to remove bubbles. Place a new gasket slide in the hybridization chamber. Load 40 microliters of the sample onto the gasket slide. Next, place the Agilent printed Virochip array with the side marked Agilent down on top of the gasket slide and fasten the screw tightly. Place the hybridization chamber in a 65 degrees Celsius oven and hybridize overnight using a slow rotation of 10 rotations per minute. To wash the arrays, first preheat buffer 2 to 37 degrees Celsius. While heating, fill a 500 ml plastic container with approximately 250 ml of buffer 1. Remove the array from the hybridization chamber, submerge the array and separate it from the gasket slide. Transfer the array to the clean container and wash in buffer 1 for 1 minute. Then, using a separate 500 ml plastic container, wash the array in preheated buffer 2 for 1 minute. After washing, slowly remove the array from buffer 2, being careful to avoid bubbles. Use a Kim wipe to wick away extra moisture, while being careful to avoid directly touching the active side of the array. Finally, Place the slide into the slide holder, adjuvant side up, and insert into the array scanner. In this procedure, we use an adjuvant 2-micron DNA microarray scanner. Scan the Cy3 labeled array at 5 microns or 2 microns according to the instrument's standard scanning protocol. Analyze Virochip microarrays by cluster analysis or the automated Virochip analysis program ePredict. Using the Virochip microarray, 2009 novel influenza H1N1 virus is readily detected in the nasal swab sample obtained from a child with influenza-like illness. In addition, we have shown that the Virochip is capable of identifying not only respiratory viruses, but also a broad range of viruses from a variety of different tissues and bodily fluids. Some examples of virus detection in whole blood and serum using the Virochip are shown here. These are ePredict calls corresponding to Virochip microarrays from blood samples, either whole blood or serum, taken from 4 patients. For each sample, the top 2 virus predictions are shown, with the top virus prediction having the lowest p-value. For example, in the sample highlighted in light blue, the top prediction for that sample is dengue virus type 1, while the second ranked prediction is dengue virus type 2. The actual virus in that whole blood sample was indeed confirmed to be dengue virus type 1, in agreement with the ePredict results. Another application of the Virochip is in novel pathogen discovery. Some examples include the discoveries of SARS coronavirus, XMRV, a gameta retrovirus linked to prostate cancer and chronic fatigue syndrome in humans, and HTCV, a novel human cardiovirus associated with respiratory and diarrheal infections in children.
Once mastered, this technique can be done in 12 hours if it is performed properly. While attempting this procedure, it is important to remember to ensure the availability of all necessary equipment and reagents. Following this procedure, other methods like specific viral PCR amplification and deep sequencing can be performed in order to confirm and follow up unusual Virochip hybridization patterns or Virochip results that suggest the presence of a novel virus.
After its development, this technique paved the way for researchers in the field of oncology to explore the association of novel viruses with cancer, in the fields of biodefense and clinical microbiology to develop condensed microarray-based assays for rapid pathogen diagnosis, and in the field of virology for characterization of novel viruses and their association with disease. After watching this video, you should have a good understanding of how to perform all steps of the Virochip microarray assay, a broad spectrum surveillance test to detect viral pathogens in clinical samples, all the way from nucleic acid extraction from clinical samples to microarray hybridization and analysis.
Don't forget that working with clinical samples is considered biohazardous, and nucleic acid extraction should be performed in laboratories rated at biosafety level 2 or higher.